Today we'll be learning about nucleotides and nucleic acids. The learning outcomes for the day, just kind of basically be able to recognize nitrogenous bases, nucleosides, and nucleotides. We'll see some structural differences between RNA and DNA, and we'll understand why there are structural differences. And then we'll talk more in detail about DNA and its structure. What contributes to the conformation of DNA? And then finally, we'll talk about stabilizing interactions within DNA and the relative importance of those interactions. Today's Meet a Scientist moment is Severo Ochoa. He won 1/2 of a Nobel Prize in Physiology and Medicine in 1959, and some of his key discoveries was the synthesis of ribonucleic acid and deoxyribonucleic acids. So let's start with some key terms. We have nitrogenous base. Here we're talking the adenine, the guanine, the thymine, the cytosine. We have the sugar component. We're either talking ribose or deoxyribose. We have a nucleoside, which is the base and the ribose, and the nucleotide, which is the base, ribose, and the phosphate. And we'll take a look at all of these things in the following slides. So here are images of nitrogenous bases. They come in two flavors, purines or pyrimidines, either with that kind of single conjugated ring or the two conjugated rings. What else can we tell from them other than their conjugation? Well, there are a lot of nitrogens going on, sometimes an oxygen here or there. Because it's a conjugation, folks, we should assume that the bases are planar in structure. Now, when we add a sugar onto those bases, we will call them a nucleoside, okay? And so to the base, we can either add a ribose that we'll eventually see used in RNA, or deoxyribose that we will see in DNA. And the difference here we can tell. Oops, didn't turn my pen on. The difference we can tell is that some of the sugars have an additional hydroxyl and some do not, the oxy or deoxy. Finally, nucleotide. So in addition to the base and the sugar, we have the phosphate added on to. These nucleotides are what we think of as the monomer that makes up the polymeric form of nucleotide, the nucleic acid, the DNA, and the RNA. These nucleotides, since they contain a phosphate, are going to be negatively charged. Therefore, we always find them associated with magnesium in a physiological conditions, under conditions, and we'll talk about this more towards the end of the series of videos. I'm assuming that many of you are already familiar with our most famous nucleotide, ATP, or adenosine triphosphate. This is because it is the energy source for many metabolic reactions in the cell, and we will learn more about this towards the end of the semester in this class. Fun fact, humans typically recycle their weight in ATP every single day. Cool stuff. Let's talk a little bit about the polymeric form. So what happens when we start building up these nucleotides together? Well, we form the polymer DNA or RNA. Nucleotides are linked to one another via phosphodiester bonds. Why phosphodiester? Well, we kind of have this, how do I circle this kind of diester arrangement about the phosphate. Each nucleotide within a strand of DNA or RNA, similarly to proteins, is called a residue. So an amino acid in a peptide is called a residue. A nucleotide in a nucleic acid is called a residue. And these residues are linked to one another, yes, through phosphodiester bonds, but through a 5' to 3' ribose linkage. What you probably will have learned in your biology classes is that you read DNA or RNA 5' to 3'. You might not have known what 5' to 3' meant, so I'm going to tell you that now because we already have all the knowledge that we need. I want you to think back to learning about anomeric carbons in the carbohydrates lecture. The anomeric carbon on the ribose is going to kind of tell us what's going on here. In our case, we have carbon number 1 as the anomeric carbon. So counting up from there, carbon 2, 3, 4, 5 makes up the ribose. The 5' to 3' language directly refers to the carbon number. So the nucleotide starts at the 5' because that's where the phosphate is, and we read it through down to the 3' carbon, which then links to the next nucleotide, 5' to 3', and so forth. So that's where the 5' to 3' originates. Now, comparing DNA to RNA is pretty straightforward. Of course, RNA or DNA. RNA is going to have ribonucleic acid. DNA is going to have deoxyribonucleic acid. So I've simplified an image of nucleic acid for us to take a look at. We see here, I've highlighted a variety of features of DNA. Probably these phosphates and the 5' ends are not going to matter because that's the same throughout. 3' hydroxyl, not very helpful at all either. This 2' hydroxyl, however, this is a differentiating factor between RNA or DNA, so this indicates RNA. Additionally, RNA has a uracil instead of a thymine, so we would again assume this is RNA. Next up, we'll be talking a little bit more about the structure and stability of nucleic acid.